Yeah, hello. Good morning, everyone. We'd like to request you all to take your seats so that we can start the program. Okay. Good morning, everyone. I welcome you all to the inaugural Dr. Kallam Anji Reddy Memorial Lecture, a tribute to Dr. Anji Reddy's contributions to science, society, and sustainability. My name is Srividya Ramakrishnan, and I head API Process Engineering. In my first few years, I have had the fortune of seeing Dr. Reddy indulging in his passion for science. It is a personal honor for me today to be given this opportunity to anchor the event. A scientist, entrepreneur, and philanthropist, Dr. Anji Reddy was among the pioneers of the Indian pharma industry. Today marks the 10th anniversary of his departure, and his legacy lives on through our work. We have a fitting tribute lined up for him today. Just to start with a couple of housekeeping things that we need to take care of, I would like to invite Mr. Rahul, a safety supervisor, to give us a quick briefing on the safety precautions.
Good morning, everybody. A very hearty welcome on this bright morning. I want to assure you at the beginning that you are in one of the safest infrastructures in the city possible. Having said that, all that we need to do in case there is a requirement for us to evacuate this place is to use the doors onto my right and your left to move out and move one floor down where there is an assembly area. Our staff will be awaiting you there to guide you further either outside the premises or to bring relief to you at the assembly area itself. All right. All the ladies who are wearing high heels, while you are moving out, please remove your shoes and walk bare feet. We have a very nice floor, and there's no need to run. There's no need to panic. Our staff will be there all along to help you, to show you, guide you, and keep you safe. Thank you very much.
And also the washrooms are also located straight down to the left. Anybody in the lobby will be able to assist you with this. To inaugurate the event, I would like to invite the family, Mrs. Samrajya, Mrs. Anuradha, Mrs. Deepti Reddy, Mr. Satish Reddy, and Mr. G.V. Prasad to the podium to light the lamp. Thank you. I now request Mr. G.V. Prasad, Co-Chairman and Managing Director, Dr. Reddy's Laboratories, to welcome the distinguished guests.
Good morning and a warm welcome to all of you. I'd like to thank you very sincerely for all of you to be here today on this very important occasion in our history. I also welcome all of you who are joining us online in the video streaming event that we are doing here. It was joyous for me to meet all colleagues from over the years who worked with Dr. Reddy and who were an important part of his life. I also warmly welcome our esteemed guest and Nobel laureate in Chemistry 2009, Dr. Ada Yonath. Thank you very much for coming here. Dr. Vijay Raghavan, a champion of science in India and a very accomplished leader of science. Thank you again for making the time to visit us and participate here. Dr. Swaminathan, Dr. Reddy's alumnus. I just called him part of the Dr. Reddy's foundation mafia. He's a very accomplished scientific leader, researcher, and a very passionate, promoter of science in India. Today is the 10th year after Dr. Reddy's passing. We wanted to memorialize and commemorate this occasion by celebrating science. And we hope to make this an annual event where we can invite and host global leaders in science and help promote the cause of science in this country. Deep science, as you all know, was an important aspect of Dr. Reddy's vision for affordable and innovative medicines. He developed the passion for science very early in his childhood. If you read his biography, he talks about the man on the moon and how it made a big impression for him. In fact, I think science is so important for a society, for a nation, and for, of course, the industry. And I think in India, we badly need this turbocharging of our advancement in science and technologies. Also, if you look at Dr. Reddy's history, the whole effort started in the lab. Dr. Reddy's was founded in a small rented laboratory space. And he wanted to, serve society through science. He wanted to make medicines affordable, accessible, find solutions for unmet needs, and this was his driving passion. And hence the title of this event, Science, Society, and of course sustainability, which is again a part of Dr. Reddy's, which he actively encouraged, though it was not a personal passion for him. He encouraged us in making the organization much more sustainable in everything that it did. So science, society, and sustainability is the theme of today's lecture. So many of you who worked with Dr. Reddy will relate to what I'm going to talk about him. There were three things that really stood up for me with Dr. Reddy the decades that I worked with him. First, he was a very big picture person, and he always thought of very high goals and ambitions, even when the company was very small. All this was driven by his passion for science. He believed that the company, the nation, we could make a huge impact if we did science and technology in a very dedicated way. Nothing gave him more joy than interacting with scientists. Many of you who worked closely with him will realize that he would go into the tiniest detail of every experiment that mattered to him. And he would call scientists at all time of the day, learn about the details, and the joy on his face when he found something innovative that happened in the lab or in the work that day was something unmatched. He would be happy the whole day about this work, and he would tell about this scientific work to anybody who wanted to listen to him. And he was always, pushing the limit on what the company could do. And he is very, passionate about telling the stories of drug discovery. I was one day baffled when my daughter, who was 5 years old then, used the word ragaglitazar. And she was just talking, and ragaglitazar is the name of a new chemical entity that was discovered at Dr. Reddy's. And it was quite a mouthful even for me, but at 5-year-old, she said, then I realized that he's been talking to his granddaughter about drug discovery. This was a very amusing anecdote that I recall. It was then over the years when we started evolving from becoming an active ingredient company to a finished dosage company to drug discovery, he became singularly focused on drug discovery. That became his complete passion. In fact, if I and Satish went to talk to him about developments in the company, on the business, on generics, he would just yawn and ask him, are you finished? And then he would tell the stories about science and the breakthroughs that are happening in the lab. And I'm sure he was talking constantly to all the scientists at the research center. So in many ways, science was at the heart of everything that he did, and today we want to celebrate that passion for science. You will also know that his title of his biography was The Unfinished Agenda, which remained his unfulfilled dream and aspiration to really discover and launch a drug globally from Dr. Reddy's. We hope that we can fulfill his desire in this decade. The second aspect of his work and his passion was bias for action. If he decided on something, he would really push the limit, push for speed, and bring momentum into everything. And wherever he was very energetic, and he would push that. On a funny side, he would call one of us in the evening and give us an idea, and said, "Do this. I think it's a good idea," and all that. And so we would agree for that. And then next day morning, again 8 a.m., he would call us and say, "What did you do about the idea that I discussed last night?" I said, "That was only 12 hours ago, and I was also asleep in the night." But that was how he was to follow up relentlessly. And he would often chide both Satish and me and saying, "You guys are spending too much time in meetings. Is this all that you do? Go and do some useful work." So he was a man of action, bias for action. The third aspect, which I think is also very important, is once he spotted talent, whether it's in the science, in the social sector, or in the management, he put his complete trust in that person. He encouraged them to take risks. He encouraged them to think big, be ambitious about what they were doing. And he was also tolerant of genuine mistakes and help people recover quickly from setbacks and look towards the future and be positive in what they're doing. I'm sure there are people in the audience who are leaders today in the social sector. I see several of them here who were flourishing under his guidance in making social impact. Also, many people who worked with him in the early days and even later on went on to become very successful entrepreneurs, scientists, and also industry leaders, and of course, creating social impact in what they're doing. So we are also very proud that several of our alumni today accompany important positions in government. The drug controller is from Dr. Reddy's. The CDRI director is from Dr. Reddy's. One of you were a part of the US pharmacopoeia. So today, I think it's very joyous for me to see all of you come here together to celebrate his passion for science. And Swami will later on talk a little bit about his experience of working with him closely in the lab. So many of these traits that Dr. Reddy had, passion for science, bias for action, trust in people, have become part of our DNA today. And we work very hard to preserve that DNA and ensure that the DNA and his legacy continues. And we added good governance to what he set up. And so science, technology, people practices, and good governance are the three pillars on which we are building Dr. Reddy's. Of course, there's an important part of him which I can't spend too much time today given the theme of today, is his desire to give back to society. There are at least three organizations which he founded, the Dr. Reddy's Foundation, the Nandi Foundation, NICE, and all three of the leaders are here today. And they become nationally recognized for the social impact. And the initial seed and the sown by him guided and mentored them to reach scale and impact. So I believe today's event is a fitting tribute to his life. The guests sitting in this room are all scientific leaders who continue to really impact the world around them. I think if he was here, he would have enjoyed being here, seeing the fellow scientists and forging those connections between industry, academia, and government. So in our own way, we hope that through this event, we can support and encourage the pursuit of cutting-edge science and technology in academia as well as industry. And through this yearly event, we also want to encourage academics, and Swami will later talk about a little bit about that. And this is our way of paying tribute to the scientist, entrepreneur, and philanthropist, Dr. Anji Reddy. So thank you again for all being here, and I look forward to the three important talks today. Thank you.
Thank you, Mr. Prasad. From growing up in a village in Andhra Pradesh to building a leading global pharma company, Dr. Anji Reddy's is a story that inspires us. It is a story that teaches us not to be timid, but to dream big. We are limited only by our imagination. What's more, Dr. Reddy showed us that it is truly possible to do well by doing good, not just for the company, but for science, for patients, and for the communities around us. Let us take a few minutes to pay tribute to the man himself through this video.
I was born in a village. I have probably seen electricity only when I was 12 years old or something. It was a dream. It was not a plan on the drawing board, no.
No.
But it was a dream. I want to be a pharmaceutical manufacturer. And obviously, God willing, maybe number one in India. If I didn't have a vision at various stages in the evolution of this company wouldn't have reached this stage. You should not think that because of failure we should cut corners or anything like that. The vision should continue in spite of the failure, because if there is that element in you which is better than many other people, you can be one of the top companies. this foundation came out of my anger. I read in Newsweek about the poverty of India, and he wrote a sentence which hurt me so much. He said, except that certain regions of India are worse than sub-Saharan Africa. I was so angry. What really drives me is the science. That can I finally bring a drug into the market which can change the way people live?
Thank you. That was a tribute remembering Dr. Anji Reddy. We will next have the keynote address by Professor Krishnaswamy Vijayaraghavan. He is an emeritus professor and former director of the National Center for Biological Sciences, Bengaluru. His most recent career contribution was his role as the principal scientific advisor to the Government of India from 2018 to 2022. Prior to that, he served as Secretary of the Department of Biotechnology in the Ministry of Science and Technology of the Government of India. Professor Vijayaraghavan received his bachelor's and master's degree in chemical engineering from IIT Kanpur and his PhD in molecular biology from TIFR. He performed postdoctoral research at the California Institute of Technology and at UC Berkeley. He started his career at TIFR in 1988 and was instrumental in the establishment of the National Center for Biological Sciences. His fields of specialization are developmental biology, genetics, and neurogenetics. His research focused on mechanisms controlling the nervous system and muscle development and consequent locomotor behaviors. In 2018, a new species of gecko was found in the state of Karnataka and was named Hemidactylus vijayaraghavani in his honor. Professor Vijayaraghavan is an elected fellow of the American Philosophical Society, the US National Academy of Sciences, and the Royal Society. He is a fellow of the 3 Indian Academies of Science and the Third World Academy of Sciences for the Developing World. He holds several high accolades, including the Bhatnagar Award, the J.C. Bose Fellowship, the Infosys Prize in the Life Sciences, and the Padma Shri. I now invite Professor Vijayaraghavan to deliver the keynote address.
Good morning, and thank you very much for having me here. It's a pleasure to be here today, and in honor of Dr. Anji Reddy. And it was really wonderful and inspiring to hear Dr. G.V. Prasad's talk and to see the movie. So Dr. Prasad, Dr. Swami Subramaniam, Dr. Satish Reddy, Dr. Ada Yonath, and everyone else. Thank you very much for having me here. I realize I'm starting a bit late, and one of my great trepidations is to delay and cut into Ada's time. So I'll be very quick and brief. But I just want to summarize before I start a few points about what I'm going to talk about. There are huge perils and promises of the future which the planet faces. And these, of course, the perils relate to climate change, energy, sustainable development, agriculture, health, and so on and so forth. And all of these, the causes are obvious or known to all of us, and I won't belabor that. But here there are promises which science and technology also has a great responsibility, both in India and all over the world. And these relate to adaptation and mitigation, and also in terms of health, healthy aging, lifestyle diseases. There are going to be new kinds of infectious diseases and dealing with that. So there's an immense social responsibility also, which again, inspired by Dr. Reddy and what he's done, all of us also have an immense task ahead. Now, India in this context is unusual. We have many lifestyle challenges going ahead. Our young demography will start getting older, and therefore problems of healthy aging, given our lifestyle, are going to become very important. India's population, although very large, is not homogeneous by any means, but it is tractable because it is composed of substantially of large endogamous groups, and therefore understanding nature and nurture together can be very useful in understanding our lifestyle and infectious disease responses. So in that context, we need to also look in terms of biotech companies and pharma at what happened a few decades ago with the growth of companies such as Genentech, Amgen, Biogen, Aguron, and so on and so forth. They grasped the nettles of early stage molecular biology revolutions and changed the way drug discovery and biologicals affected our health. Today, I would like to stress that there's another opportunity, and that opportunity is not just for laboratories and companies in the West, but also for laboratories and companies in India. And I will use the brain as an example to show what these opportunities are. Basically, what I want to say is the following: that because of evolution, and because evolution links all organisms on Earth with a common origin, and therefore a shared chemistry connected through the thread of DNA, studying any organism can impact on studying any other organism. So far, multiple therapies have come through by this conserved molecular and cellular aspect. Now there's a great opportunity for looking at organisms as a whole, to looking at how they function, tissues and organisms, and that is the new opportunity. And I will use brain as an example to show how studies from model organisms to cell culture to humans can be the new age of discovery. But this is true for any tissue and any kind of problem. Okay, so we heard about Dr. Reddy, and again I'd like to acknowledge his wonderful contributions and also point out that this should be an inspiration for us to link academia and industry, the public sector and the private sector, much the way in which his life itself was. So the brain in health and illness. Oops. Now the human brain has 86 billion nerve cells and trillions of connections between these nerve cells. And therefore it seems its analysis in health and illness is an impossible task. How do we study a tissue of this size, which has completely changed the way we live and also shaped this planet? Now, the brain is susceptible to both problems of development, genetics, environment, and infectious disease. And here is a brain infected with a tapeworm and causes neurocysticercosis. And this is a widespread problem, and this is an example of how brain diseases can have a great impact. And these were treatable by the development of technologies. I won't go into that now, but there are examples, other examples too. Other diseases, such as Parkinson's, schizophrenia, depression, and so on, have other causes which are poorly understood, a combination of inherited and environmental causes, and their study also is a great challenge. Now, if the brain has 86 billion nerve cells, and we want to understand these diseases and we want to understand it in health, how can we do that when our understanding of a bacterial cell, shown over here, which senses temperature, pH, sugars, carbon sources, and so on and so forth, right? Our understanding of this bacterial cell is still not predictable. We understand how when it receives a sugar cue or a temperature cue, how it can respond. But when all these are put together in real life, as it were, our understanding about predicting all of this is very low. So if we if we can't deal with this, how can we even deal with 86 billion cells? Yet, as I said, there's a connection between evolution which allows us to do this, and this complex brain can be broken up into small parts. By the way, this is a blank slide, so normally when I show a blank slide, the IT people rush and try to see if something is wrong. There's nothing wrong. Yeah. The challenge is that the solution is that the brain can be broken up into components, into units, its nerve cells, its regional specialization, its chemistry, and therefore a lot can be understood even though it's enormously complex, much the same way in which we do for a bacterial cell. So this question is always asked of biologists. Where does it come from? That's evolution. Then how is it made? That's developmental biology. And how does it work? That's physiology. And biologists only ask these three questions again and again and again, no matter what they're working. So let's ask where it comes from. We came from, and I'll quickly run through these slides in the interest of time. We have 1 billion years of multicellular life on this planet. We have 400 million years of insects and seeds, and this is the way we came across to that position. And we have 130 million years of flowering plants, 200 million years of mammals. And then something dramatic happened 65 million years ago, and this shows how things can change. And we are here because of an accident, and that of an asteroid, a meteorite actually, crashing on Earth near Mexico, and that wiped out large dinosaurs and resulted in the growth of the mammalian radiation. And then it's only 25,000 years since Neanderthals, cousins died out, and we mated with them, we mated with Denisovans, and we have their DNA. So this dramatic change which took place 65 million years ago allowed for accidents of evolution to take place again and us to come about. In that situation, let's look at how the brain is made, or how any tissue is made. And this slide is from a friend of Ada's and mine, Benny Shiloh, who runs a course in Bangalore every other year. And basically it points out that whether you're a fly or a worm or a mouse or a human, the toolkit for making you is the same. In other words, if you're making a small aircraft or a huge big one, nuts and bolts and screwdrivers and the aerofoil and the engines all are used, but they're used in different ways in different contexts to make different organisms. Because the toolkit is same, we can learn a lot about studying any organism by studying any other organism. This is not just a concept of evolution, but in molecular terms, this has been shown to be true in practice. So on the top is the embryo of a fruit fly, on the bottom a mouse, and many genes are conserved not only in the way they work, but in their patterns of expression. And in terms of the brain, the principle is very simple. On the left is shown how brain cells develop. They develop from stem cells, which proliferate to give rise to more stem cells and to nerve cells. So these are the nerve cells over there. And these then proliferate along the axis of the animal with different kinds of properties depending on where across the animal these cells are born. So position of where a nerve cell is born specifies in many ways its identity. So this is a simplifying feature. Just as position is important, timing is also important, and when a cell is born is also important. And that you don't need to, I won't get into the details of this, but the diversity of the nervous system comes from timing and spatial position, and both of these result in the enormous diversity which we have from brain to spinal cord to our arms and legs and so on and so forth. So that's how it's made. How does it work? Well, again, evolution comes to our rescue, and Hodgkin and Huxley studied the giant squid, and by just removing out in the long nerve cell axon its contents and poking electrodes into it, came across discovered beautiful biochemical principles and predicted how channels can open and close and allow ions to flow in and out just by looking at physiology. And this is an amazing development, an exquisite example of modeling by doing physiology and predicting what the molecular properties were, not by studying any molecule, and all of that happened later on. Now, you can study one cell. Here are 20 million cells, Purkinje cells in the brain. How do you understand 20 million at a time? Well, in a manner similar, but a great advance over the Hodgkin and Huxley method, you can now record simultaneously thousands of nerve cells at a time. You can do that in dishes, you can do that in open brain preparations in model organisms, so one can get an idea, and we still don't know the meaning, though, of many of these recordings which we're doing, and that's a major area of research. So large-scale understanding of how many neurons together work is a major area of research. On the left is Ramana Kahal's drawings of the Purkinje cell, and this is an actual picture. Now, nerve cells, our brain is not just a jumble of nerve cells, they're connected in a specific manner. And this is a drawing from Ramana Kahal of connections, and this is a schematic showing those connections, how signals go from one end after receiving signals from other neurons, go across an axon and go to the other end. Now, this kind of drawings which Kahal made were very important, but now scientists can look at nerve cells on very large scale. And this is a drawing of a nerve cell in the fruit fly Drosophila, and scientists do EM electron microscopy reconstructions of parts of the brain, and now entire brains, align these sections precisely and reconstruct them in this manner, and look at connections also between nerve cells, and have developed the connectome of a maggot of the fruit fly of about 3,000 nerve cells, and soon the entire brain of the adult fly, which is 20,000 nerve cells. And now they're working on 1/500th of a mouse brain, which will take several years to get a connectome off. With rapid development in electron microscopy technology in parallel processing and alignment, even though at current pace it seems as if a mouse brain will take decades and a human brain will take hundreds of years to get a full connectome, it's quite possible that dramatic changes in technology will let this happen much, much faster in a manner similar to how sequencing technology, DNA sequencing technology, changed the pace of generation of DNA sequence data. So it is reasonable to expect, just as at the onset of the DNA revolution, that there will be dramatic changes, and companies went forward and tried things which seemed impossible and seem straightforward now. Similarly, there's a great opportunity in brain research and other areas of research because of these dramatic changes in technology which are forthcoming and which are happening now. This annotation and proofreading was done both by computer and by hundreds of students, undergraduates and graduate students working, and these very careful segmentation analysis and proofreading results in high quality, and this results in a complete connectome of a fly brain, as also I told you about the fly's maggot. What use is this? Well, you can now look at a freely moving fly or a tethered fly, record from any neuron, activate any neuron or turn it off, look at the behavior, put drugs, see what happens, alter it genetically and see what happens. Now you can not only do this in the fly, but you can actually do this in the brain. You can do it in a human brain or a mouse brain by taking induced pluripotent stem cells, taking them along differentiation pathways, and making them develop into different parts of the nervous system and analyze these parts of the nervous system by physiology, by anatomy, by putting drugs on them, and so on and so forth. So there's a great opportunity to directly work on human cells of different types, both to test drugs and their effect, look at various genotypes and their consequences, and for actually looking at how circuits function, in addition to looking at whole animals from mouse to human, for example. So this brain, which seemed impossible to break up and understand, is increasingly becoming susceptible to analysis, and this will only get better as computer science, mathematics, statistics, and biology all come together on large scale. There's an enormous opportunity in countries like India where these areas of research, mathematics, computer science, the handling of large data, is something which school children, college children, teachers are very passionate about, combining that with biology in a manner which is accessible and practiced in many laboratories in India. So there's a great opportunity for entrepreneurships and for industry to have an impact. So is the human brain special? We know that in terms of brain size, the human brain is large, but it's not the largest brain. Elephant brains are larger. But it looks like with 86 billion neurons, the human brain has the largest number of neurons. People haven't actually tested whether the elephant brain has got more neurons, but it looks like the elephant brain has got more glial cells and not more neurons. So 86 billion neurons make the human brain not special in terms of molecules, not special in terms of the kind of small circuits, but special in terms of the extraordinary parallel processing which such a large number of neurons tiled across different layers of the brain do, and that has resulted not just in biological evolution, but cultural evolution of the kind we have seen. So this is the elephant brain, and we don't know about that yet. So I'd like to end by saying bacterial cells are complex, every other kind of cells are complex, but complexity is something which humans have become good at cracking, and while these tasks are, the task of looking at the brain is enormously difficult, it seems a problem which is being attacked by several people, and it's also one of great opportunity for those in India. So thank you very much.
So thank you very much, Professor Vijayaraghavan. I think that was truly inspiring on how you can look at, unravel the complexity of the human brain by understanding, starting with the fruit fly. So thank you so much for the very inspiring keynote address. I would like to invite Mr. Satish Reddy, Chairman of Dr. Reddy's Laboratories Limited, to present a memento to Professor Vijayaraghavan. Thank you, Satish. I now request Dr. Srinivas Uruganti, Director of the Dr. Reddy's Institute of Life Sciences, to now introduce our next speaker.
Thank you, Srividya. It is my privilege to introduce Professor Ada Yonath, who will be delivering the first Dr. Kallam Anji Reddy Memorial Lecture. Professor Yonath was born in Jerusalem, Israel. She graduated from Hebrew University, earned PhD from Weizmann Institute of Science, and subsequently carried out a postdoctoral research at Carnegie Mellon University and MIT in the US. Since 1989, she is the director of the Helen and Milton A. Kimmelman Center for Biomolecular Structure and Assembly at the Weizmann Institute of Science. During the period 1980 to 2004, Professor Yonath headed the Max Planck Research Unit for ribosome structure in Dessau, Hamburg, in parallel to her activities at the Weizmann Institute. She is the recipient of the 2009 Nobel Prize in Chemistry for her pioneering studies on the structure and function of the ribosome. She is also a recipient of many other distinguished awards: European Crystallography Prize, Harvey Prize, Wolf Prize, L'Oreal UNESCO Award, Albert Einstein World Award, and the Marie Curie Medal. Professor Yonath's research focuses on genetic code translation of ribosomes, antibiotics which paralyze this process, designing novel eco-friendly antibiotics for fighting resistance, ribosomopathies, and also focused on origin of life. Professor Radha Yonath will be speaking today on her lecture title would be From Origin of Life to Next Generation RNA-Based Therapeutics. Let us extend a very warm welcome and a big applause to Professor Radha Yonath.
Hello, ladies and gentlemen. It's a big day for me to be part of memorizing Dr. Reddy, as his contribution is much over than the normal one of scientists and also of business people. He was really the most suitable mix of both science and business. I'm sorry that I'm not able to talk to you about the two aspects. I'm just a scientist. But I have something to tell, and I hope that you will like to hear it. So, essentially, I will describe what we are doing in the last few years, which is, of course, based on the pioneering studies that we made in the past. So essentially, I will talk about the origin of life, from it to the future of antibiotics or to future antibiotics, and also I will touch on genetic diseases. So the common subject of everything is the ribosome. And I want to tell you the ribosome exists in every living cell. And you saw some examples in Vijay's lecture a few minutes ago. Ribosomes are made of two subunits. The small subunit is where the decoding of the genetic code comes. So maybe I should start differently. I should start, ribosome is the position in all cells where the genetic information is being translated to the products, the proteins. The genetic information is brought to the ribosome on messenger RNA, which is a copy, more or less a copy of the DNA with the original genetic information, the cellular one. Now, when I say messenger RNA now or mRNA, everybody knows because of the vaccination, the COVID vaccination, but actually it's a very important short-lived copy of the piece of DNA that has to be translated at a special moment. So the messenger RNA is made of nucleotides, and each 3 nucleotides have a matching another molecule of RNA, transfer RNA, which knows to connect to the triplet that is chemically connected to it, or chemically can be connected to it, and an amino acid that is specific to this triplet. And there are enough triplets for all the 21 amino acids. So the messenger RNA reaches the ribosome, actually reaches the small subunit, and is located in 3 sites, A, P, and E. A, amino acylated, P, the peptidyl, where the protein will grow, and E, the exiting one. The bond will be made here in the large subunit between the 2 tRNAs in A and P positions at the active site, and the newly born protein will come out of the ribosome through a tunnel. And I'll show it in a minute in a little bit more detail. But before this, highly active mammalian cells, like the liver, for instance, may contain up to 6 million ribosomes in a single cell. Even in bacteria, and we will talk a little about bacteria because this is the position of the infectious diseases, up to 80,000 ribosomes in one bacterial cell can function, and very efficiently. So when we started the work, and when we got the first structures, we made a little movie together with art students from the Art Institute in Jerusalem, Art Academy in Jerusalem. And I want to show it now because although it is over 20 years old, this little movie, I think it explains almost everything needed to understand our work. So let's see if it works. Should. We made it with 2 students from the, as I said, from the Art Academy. One of them became in love with science and became a scientist instead of artist. So what you see here, the DNA, actually, the messenger RNA, is reaching the small ribosomal subunit that you can see here. It goes into its path, which is directed by initiation factor. There are initiation factors even in bacteria, 3 of them, in human more, and this controls the beginning and also the accommodation of the first tRNA according to the sequence, which comes with the amino acid. You will see the amino acid, you can see it now, this very little piece, which is the most important thing. This will become the protein. So the large subunit is now reaching and making bridges to the small one, and the ribosome is ready to work. Now more and more tRNAs are coming with elongation factors, and they come into the ribosome according to the sequence. The ribosome helps in, out, in, out, in, out, and the reading is done, the reading of the sequence is written in the small subunit, the genetic code, and in the large subunit, which we took away now, that you can see that the interactions are between the pieces that are bound to the other side of the tRNA. So you see the whole ribosome again, and protein starts growing and starts coming out through the edge of the tunnel, and they come out more and more as long as there is a code. And when there is a stop codon, it means the end of the code, then instead of tRNA, release factor and recycling factors are coming, and the protein is being made. It can now come out in here at the end of the ribosome, and the rest of the participants are ready for the next job. And they can do it very many times in a very short period. That's all. That's the way proteins are being made by the ribosome. In the previous slides, you saw the ribosome as, can you leave the darkness, please? Thank you. You could see the ribosome as one piece, but actually it's made of many, many components. Most of them are ribosomal RNA, which is shown here in gray. Actually, you see here the small subunit and the large subunit. The ribosomal RNA, over 70% of the ribosome is made of it, or most ribosomes, and ribosomal proteins, which are shown here in different colors. Just to remind you, the small subunit is responsible for the decoding, and the large one for making the peptide bonds. So if we look at it from the way that the ribosome is connected, here is the small, here is the large subunit. tRNA is reading the sequence here and producing the bonds here in the large one, so this is tRNA. And the other positions that I talked about, messenger comes in here, and the decoding center in the small subunit. The position of peptide bond formation, which is called PTC, peptidyl transferase center, is in the large one. That's the whole thing, and we can look at it also from this side. Small and large subunit, and the 3 tRNAs, and the position of the decoding and the PTC are shown here. When we look at the PTC in more detail, we see, or actually we saw, that it's here in the large subunit, and it's made of 2 parts, the part of the A site is in blue and the part of the P site in green, and I will keep this color in the next few slides. They sit like that, like a pocket, like semi-symmetric pocket. And if we want to look at it in terms of atoms, this is the position. And this is fully conserved in all known ribosomes. And even in those that were not known in the beginning, by now there are many more structures because the technology became much more available, and they are all the same. And I just want to remind you, we look at tRNA molecule. It has an anti-codon loop here. Here it reads the sequence, and at the end of it, where peptide bond is being made, where it is involved in making peptide bond, it's always throughout evolution, from the smallest to the largest cells or animals, it's always CCA. Cytosine, cytosine, adenine, and amino acids, always. So the structure is very similar. The chemistry is the same. What does it mean? Actually, if we look at it from here, it means something more than just being the same. It means that there is no straight enzymatic activity. It's positioning rather than little chemistry. So if we look at the piece going from the A in the blue to the P in the green, it is going through through its position in the ribosome according to the path provided by the ribosome, which is here in red, and makes all these contacts with the ribosome while rotating, going from A to P. And this happens to every amino acid that goes into the protein. So all this led us to propose that there was a protoribosome that is highly conserved in the symmetrical region, and this indicates that peptide bond formation is beyond the existence of environmental conditions. It's there. It became there. We don't know how, but we think it became. So when we look at what we think is the protoribosome, our concept is it sits in the large subunit. It has two parts, blue and green, for the interaction of the tRNA and the position where it comes out. It looks from the side like this, like a wonderful pocket. And if we look at it in many, many different ribosomes, it's the same. Here we drew just the main chain of 6 different ribosomes from 6 different organisms. They are the same. We can't distinguish between them once we drew them. If we look at it where it comes from, the structure of the RNA, it is based on this piece, which is the piece that is important for peptide bond formation. And it has 2 parts. The top part, which is where the P site tRNA binds, and the lower part where the A site. They, of course, sit like a pocket, but these are the components, and they are the same in all ribosomes except for these additions, which probably came out after there was a protoribosome. So once we look at the protoribosome with a little bit more detail, you can see it rotating with the substrate inside and the RNA components of it that are shown here, the A and the P in green and in blue. We decided to produce this piece of protoribosome in the lab. It took 10 years to get relatively stable pockets. And I can show you here how we produce them from the chemical point of view. I really don't want to go into detail unless you ask me. But I can also tell you that some of them, not all of them, were capable of making peptide bonds. Just as them, without ribosomes, without any code, without any sequence information, nothing. This took another 5 years and actually was achieved by an Indian postdoc that came from India, Tanaya Bose, who found a way to show all these five examples, and now she could show more. So what does it tell us? It tells us that from the protoribosome to the modern ribosomes, the sequence of each component of the central region has been genetically optimized for the functional task. In the beginning, they were the same because there was no difference between entrance or exit. It was for the pocket the same. But later, the A site, which is entrance, and the P site, which is an exit, had to be modified for their needs, or had to be engineered for their needs. And this story that excited us very much excited also nature, and just 2 weeks ago, we got these pictures from them after they interviewed us. They think that the ribosome was a mixture of many, many RNA chains. From them, a protoribosome came out, and later on the whole ribosome, which is blue and green according to our colors, and the structure that would be developed into ribosomes. So that's my telling you about what we know now, or what we think we know about the protoribosome. But it's not the end of what I want to tell you. I want to talk about further development, and this is the ribosome as an ultimate target for antibiotics. And actually it is. The ribosomes are, because of their fundamental role, they are the target of over 40% of the clinically useful antibiotics. Almost half of the non-antibiotics, and no matter if they were made by a big or small company, by somewhere here or there, they are targeted by antibiotics, and very successfully. So how do the tiny antibiotics paralyze the giant ribosome? So the ribosome's molecular weight is over 2.5 million Daltons, and the antibiotics are usually less than 1,000. A huge antibiotic is a little bit more than 1,000, but it's nothing compared to the size of the target, the ribosome. The antibiotics are really very clever. They target important regions in the ribosome. So what you see here is a section through the ribosome, tRNA in the P site, the newly born protein coming out from the protein tunnel, and erythromycin, which is the first antibiotic for ribosomes that came into use, is just blocking the tunnel, and I'll show you more detail in a second. But before this, I want to show you that all the antibiotics that target ribosomes target important regions, functionally important regions, like the decoding, tetracycline, like the tunneling, erythromycin, like the bonding and the hinging, and I will show you one by one. Actually, all these names you may know, but hinging is the part of motion that has to be made in order to open and close the path. And the names of all the antibiotics that I'm referring to are shown here. So let's look into the tunnel. It's the large subunit, and the entrance to the tunnel, and erythromycin blocks it. It doesn't block it fully, but it blocks it enough not to let the newly born protein coming out of the ribosome. So it stays in the ribosome, it means the ribosome is not useful anymore. So we made also a little clip, little movie about antibiotics that target the ribosome. And I want to show you, it works here. 4 of them. The first one is edein, this yellow, and edein was found in Poland. It's not very useful because it's not very distinct, it doesn't distinguish very well between the bacteria and the patient, but it's very illuminating to understand how they work. Just doesn't let the messenger bind. That's it. Very clever. Very small, very clever. Even smaller, tetracycline, which occupies the position of the A site tRNA after the P site is already bound, or the next tRNA after there is already some binding. That's it. No binding. The third one I already mentioned. It sits in the tunnel, erythromycin, and it is in the large subunit, just blocks it, blocks the motion through it. And the last one that I want to show you is chemically the most clever one, goes into the bond. It disturbs the formation of the bond, of the peptide bond. So it will come here, choop, in the bond. That's it. So we were very excited by understanding how antibiotics work, but we are still fighting, and the whole world is fighting with resistance to antibiotics, which is one of the most severe problems in modern medicine. How does it happen? So let's come back. We're looking here. This is the large subunit. Here is the tunnel. Erythromycin that I showed you earlier is here. It means it blocks, if we look at the tunnel like a line, a vertical line, it blocks it. And not only it, the whole family of macrolides, which is the family of erythromycin, block the tunnel. Either they block excellently, or just very good, or just good, but doesn't matter, they block. And all of them interact with one of the tunnel components, which is adenine in position 2058. Here, this pink one. And this is correct for all bacteria. It's always 2058, adenine in 2058 in all bacteria, in all ribosomes. But not in human, not in higher organisms. There it is not A, but N. So this is the situation between erythromycin in the tunnel and the position 2058, which is adenine in bacteria. And here the contacts are very good. It means erythromycin will paralyze this bacteria. What will this bacteria do? It will imitate itself to human, make itself G. From A to G, this is the procedure or the strategy that the bacteria takes, but when there is a G here, the distance is too short and there is no binding. That's so simple. This is resistance, the simplest resistance. Start with A, N mutation, maybe one of million mutations, but this mutation is suitable and wins. That's it. There is a resistance here. Can we combat resistance? Can we think about combating resistance? I'm sorry, I don't have an answer for that. So far, we are not successful. Not only we, even the greatest companies. And you can ask Dr. Vijay. Resistance to antibiotics is a very tough, very, very tough assignment. But actually, before the bout, most pharma companies stopped searching for new antibiotics because they're afraid that there will be resistance and then their profits will drop dramatically quickly, or relatively quickly. So what do we do? First, have a look at what the companies are doing. On the left side, you can see the growth of resistance of 3 different bacteria in 12 years, from 1990 to 2002. On the right, the new antibiotics all over the world approved in 30 years, from 84 to 2014. That's it. Not even 10. Isn't it a big problem? Actually, pharma companies are not making new antibiotics. Most of them are not making new antibiotics now. They are refining or producing more sophisticated antibiotics from the older ones. Like these are the macrolides, and here are all types of tails that they bound to it. But this doesn't work for a long time. It works, but not fully. So we decided to look for a different way of producing antibiotics. We look for next generation, but we take advantage of our knowledge, and I'll show you in a minute why we think so. We can make environmental friendly and some other positive characterization of the newly born antibiotics. So that's what I said just now. What you see now here is the main chain, the skeleton of the large ribosomal subunit from a normal non-pathogenic bacteria in gray, and little pieces that were found additional on a pathogen. Just these little bluish things, which are actually Staph aureus. So these ones, they are all existing only on the pathogen, and they look like this. For instance, in the neighborhood of this one, all the neighborhood is the same, except for this piece. So this piece is specific to Staph aureus, and it can be a potential binding site, in our opinion. And there are many more like this. This is what I showed you until now, but actually there are many more, all this, and even more than this. So this can be new anchors for new antibiotics, and we can look at it, you can see it also when we look at the sequence. These are sequences of many bacteria. Staph aureus is on top, and it's the only one that has here a piece that does not exist in this. So we can target this one. You can align and target this one. You can see it here too, or here, or here, or here, or here. I just wanted to show you that there are many. I don't want to go into each detail. And we found that we can inhibit protein biosynthesis by targeting these positions. You can see here the number of nucleotides, that's the way we are counting them, how large they are, and where they are in the place of the ribosome. And we can also show the size of what we need in order to target this particular RNA piece. It doesn't have to be RNA, and we can target them differently, but this is an example. Before I finish, I want to tell you that looking at ribosomes, we can also look at genetic ribosomal diseases. And this we do together with my doctor, Dr. Yonat, who is here, who is an MD, a geneticist that works, runs also the genetic institute at her hospital. So about 3 years ago, there was a paper showing how many differences were found in ribosomal genes of different cancers. And it's 25% of the cancers. Also, before I go, sorry, I want to talk about one example of them, which is ribosomal RNA 1284, which was shown to be associated with inactivating or disturbance of the p53 control mechanism, and that's the way we think it works. We also know, and this is actually Hagit's work, that there are 97 different mutations, inherited mutations, in all body genes of a disease called DBA. It's an anemia, and we are trying to understand mainly 2 questions, but not only. Are the mutations expressed in the ribosome or only in the genes? Because all these mutations were found so far in the genes. And why there is a disease? What are the reasons? This is still not known for DBA and also for the cancers. And if I want to relate to Vijay's lecture, also in degenerative diseases. What we do know is all the positions that we found, or not only we, that found differences in genes, in ribosomes, in cancer, and in DBA, are in proteins or around proteins that are on the surface of the ribosome. It means there is a very good chance that if they are expressed, we will be able to target them. So in short, we are creating a novel, universal chemically controlled platform for identifying, characterization, and targeting mutated ribosomes in diseases that are called ribosomorphomatis. Before I finish, I want to thank the Weizmann Institute that let me work, although I was considered a cuckoo, a dreamer. The Weizmann president, who died about 3 or 4 months ago, Professor Sela, made the decision. He was a biologist, but also a physicist. Professor Harari continued. I was encouraged by the Scientific Advisory Committee, Professor Kendrew, Anfinsen, and Rich. I'm sure you know all these names, and was highly supported and had very, very beautiful collaboration with the Max Planck Institute for Molecular Genetics in Berlin, headed by Professor Wittmann. I had 2 research groups, as you heard, in Germany and in Israel simultaneously, and I want to thank the 2 group members. I cannot show you all of what happened the whole years, but what happens in my group now, the contact with Max Planck terminated almost 20 years ago, and this is the group in Israel. Actually, some of them are already don't belong. Those that belong are with these little sons. Among them, I want to highlight Adlat Bashan, who is the head of the group, although formally I am, but she is doing the whole work and the whole supervision. I want to highlight Tanaya Bose, the postdoc who were able to show that protoribosome can work without ribosomes, without life around it. I want to also highlight 2 other Indian students, actually 1 student, Disha, and 1 postdoc, Rajan. So this is my little Indian corner. And I want to show you Tamar, who came 20 years ago for 10 weeks. She's still around, and she had a birthday the day that I took the picture. She made a cake. You want to see her cake? This is her cake. And this shows that in my group, ribosomes are considered sweet. I also want to tell you that Hagit, who is my daughter who is here, and her daughter, Noah, are very supportive. I want to thank them. And they are so, although I disappear many times. And I want to tell you what my granddaughter, when she was 13, she said. As you know, she, it means me, is a very busy scientist, but she always finds time for me. And she gave me a prize of her own. That's the Nobel. This is her prize. For me, more important. The grandma of the year is Ada Yonat. And she told me that she will never take it off the wall. And indeed, she did not. On the condition that I am still the best grandma. So probably I am. Kids are celebrating the Nobel. in carnivals, and Michel Kishka drew me as head full of ribosomes. Thank you. Yeah, sure.
So, thank you, Professor Yonath, for the wonderful lecture and also highlighting the problem of antibiotics and AMR. But thankfully also you have shown the potential way forward and the pathway which is possible to adapt and work with the bacteria which are quite tricky on their own. So we will have maybe time for a couple of questions. I think it's a great opportunity if somebody wants to ask. Maybe a couple of questions. Yeah, Professor Sharmishta. We'll get you the mic, please.
Wonderful listening to you. But what I also understand is this does not matter what molecule we are going to make against a bacteria, evolution is a process. So eventually they may be overcoming it. In that context, the recent idea of a host-directed therapy along with using antibiotic, how useful is that going to be, if you can comment on it?
I'm sorry, I didn't understand.
How useful can be a host-directed therapy along with the approach that you have indicated, both together to fight the...
If it works? If it works, wonderful. I didn't think about it yet, so maybe you won't.
Thank you for a very brilliant lecture, and thank you for being in Hyderabad. I have one question for you. I'm a physician, and I deeply wedded to scientific discoveries. Now, we have seen few decades in the last century that human disease was focused on mitochondriopathies. There's a lot of work that went into that mitochondrial structures were responsible for many of the neurological disease, degenerative disease, et cetera, et cetera. Now, your revealing work, which is absolutely masterful, we have focused on the pre-ribosome concept and introduced the term ribosomopathies. Should we anticipate that these are all kind of great discoveries, but ultimately we're going to transition to something else we're going to find as a process of evolution and scientific discovery? Or are you confident at this point in time this is truly the secret for the human being and we're going to have these massive discoveries from your work?
Thank you for the idea. Mitochondria is really a target that was less identified, even for the ribosomes, although many of the problems of neurodegeneration is in the mitochondria. And we started to look at it recently, really recently, this year. So hopefully something will come out. I know that we are not the only ones. I didn't want to say that since we are looking at it, this is, there is an answer. But I hope that there will be some type of answer, because the way antibiotics, anti-cellular and cellular problems are being looked at a bit more, wish to cure them. So maybe there will be something that will come out.
I think, so let us all once again thank Professor Ada Yonath for this illuminating lecture. So I will now request Mr. G.V. Prasad to hand over the to present the memento to Professor Yuna.
This is yours. We have
No,
to.
this photograph is. Maybe here. Yes, we'll have a photograph. Yes, that's
Somebody took it.
Thank you very much. Our next lecture will be by Dr. Swaminathan Subramaniam, CEO of Ignite Life Science Foundation, a non-profit organization that curates science to amplify the positive impact of research. Dr. Swami is a physician scientist with an MD in clinical pharmacology from the Postgraduate Institute of Medical Education and Research in Chandigarh, and a PhD in molecular neuropharmacology from the University of Pennsylvania. He was an NIH Fogarty Fellow at the National Institute of Neurological Disorders and Stroke. He has won a Gurkha Shevning Fellowship for Leadership Excellence at the London School of Economics. In a career spanning over 25 years in India, Dr. Swami has held several leadership roles in the biopharmaceutical industry and in healthcare companies. He was instrumental in Dr. Reddy's foray into providing pharmaceutical services and also served as the Managing Director and Chief Operations Officer of Origin Discovery Technologies. He led the filing of the first investigational new drug by an Indian company for a novel topoisomerase inhibitor as an anti-cancer drug. He is also the author of two very interesting books titled Mastering Sleep and Healing Hands. I now invite Dr. Swami to come onto the stage for his next lecture.
Thank you very much. I will start. First of all, Professor Yona, that was a brilliant lecture. What I took away from that lecture, even though I'm a scientist, the message that I took away is one video is worth 10 slides. So if you can convert your hard to understand stuff into a video, it's always better. I really enjoyed those slides. The second thing I'm sure we all took away is that science is very complex. A lot of that was intellectually quite challenging to do that work. So what the corollary from that is we need our smartest people to work on science, right? We need the most curious, most intelligent Indians to be working in science. The rest of them can go to other fields of endeavor. The other thing you would have noticed in the last slide she showed where she showed her team is science is a team effort. The days have gone when there was a single scientist laboring in a lab making a discovery. It's now completely a team effort. it's not simply a team effort, it's an international team effort. There are international postdocs, international scientists, international collaborations, and global science teams working on problems. So I think we have to keep these things in mind when we think about the future generations of scientists in India. And I'll be talking about a few of those things. I'll start by thanking the hosts for this morning, Prasad, Satish, Dr. Reddy's Laboratories, for inviting me here. I am truly very privileged. I'm privileged in two ways. One, I was privileged because in some senses, I worked very closely with Dr. Anji Reddy. So that is one level of privilege. I'm privileged again now that I'm invited to speak at his memorial function, which we are all participating in. I'll start by saying that my association with Dr. Reddy's group probably started sometime in 1993. And I was not headhunted, I was not recruited. I actually headhunted Dr. Reddy's because there was an ad for chemists in India Abroad. I don't know if some of you may know of India Abroad, which is a broad magazine in the US. And there was an advertisement from Dr. Reddy's for chemists. And I said, if they are hiring chemists for drug discovery, they must need pharmacologists at the end. So I applied for the jobs, and they did not come looking for me. I went looking for them. And several months later, I got a reply, and I was interviewed twice. In the first interview, I think it was Dr. Venkateshwarlu, who interviewed me at a Courtyard Marriott near where I was staying. And I remember this very vividly, that I went to the place where he was staying, and he made me wait in the lobby for quite some time. Or maybe there was some kind of a strategic tool to say that you are coming after us, we are not coming after you. But anyway, I persisted, and my second interview was with Prasad. And Prasad asked me a question in pharmacology, and I think the company was working on ibuprofen, and he asked me, how do you screen for an anti-inflammatory? If I had been an NIH scientist, I would not have known the answer to that question. Fortunately, I had done my MD in clinical pharmacology at PGI, where those are the things we learn. And I think I answered his question to his satisfaction, and I got this wonderful job at Dr. Reddy's, which has been one of the best experiences in my life. So this 10-year milestone is a good time to look back and reflect and examine the meaning and lessons his life, Dr. Reddy's life, carries for us now. And honestly, I speak from, I left the group in 2002, a decision, I wouldn't say I regret, but it was a difficult decision. It was a great company, and I was leaving. But that means that my memories are at least 20 years old. So, but it's not a bad thing to have memories fade a little bit. Because for this reason, if you take a paragraph of text, and if you remove all the punctuation marks, the text brings out some new meanings, right? In a similar way, when you have memories, old memories, and many of those trivial day-to-day incidents have faded from that memory, what you're left with is the true memories, the true profound truths from your past experience. So I think I will believe that when I speak today, I speak from that kind of a situation. Reading, and the other thing I must admit is that apart from my own memories of my experiences, I was also benefited by reading and pondering Dr. Reddy's excellently crafted biography, The Unfinished Agenda. It gave me room to revisit my thoughts about him, and really that's what I'm going to talk about today. First, I would like to again take you back to 1995, the year I joined Dr. Reddy's. Although I accepted, I was interviewed in 1993, it took me 2 years to join, because I think things were not quite ready at this end. So in 1995, I joined Dr. Reddy's, and there was a certain mood in the company at that time, and we all had that mood. And if I had to use a word to describe that mood, I would use the Yiddish word chutzpah. And I discussed this word with Ada Yonath and her daughter last night and tried to get to the real meaning of the word in Yiddish. But the English translation of chutzpah is ambitious. And even that literal translation, I don't think, does justice to what chutzpah really means. And if there are no millennials in this room, but if there were, I think they might understand that chutzpah means something like ambition with swag, right? So I think that is what percolated through the company at that time. And I think this came from the top, Dr. Reddy himself. It came from his ability to dream of a destiny for Dr. Reddy's laboratories that would not have seemed reasonable or possible within the frame of reference for Indian companies at that time. This feeling of everything is possible for us spread from Dr. Reddy himself throughout the company. We all gloried in it. Dr. Reddy's confidence was not one of the empty kind, inflated by bravado. It was born on his self-confidence and analytical intellect, sharpened by his incredible academic background. His PhD advisor at NCL, Dr. Duraiswamy, was a giant in the field of chemical engineering and was ranked among the 10 best chemical engineers in the world at his time. And he is quoted saying this about Dr. Anji Reddy, start quote. Indeed, I have rarely taken a better decision in recruiting students and even scientists. He not only produced a very fine thesis, but also worked on a side problem to produce a paper in a matter of weeks, which to this day is quoted in practically all books and reviews on properties estimation. The Reddy-Duraiswamy equation, developed by him, Dr. Reddy, with little assistance from me for predicting liquid diffusivities, has lost none of its original flavor. End quote. This paper was published in 1967. I checked on how often it is being cited now in 2023. Since the year 2019 till today, it has received 55 citations, which is incredible for a paper of that vintage. From his PhD, Dr. Reddy moved on to become a business person. After a stint at IDPL, as an employee at IDPL, he became a pioneer in the bulk drugs industry in India. And as a founder of a bulk drugs company, Dr. Reddy combined the problem-solving aptitude of a technocrat with the pragmatic attitude of an entrepreneur. His early success was rapid, but by no means easy. After initial hiccups, the company grew quickly and profitably in the technology-intensive bulk drugs business. This success was tied to R&D. Science, when done well, is almost like magic. Dr. Reddy, already a good scientist, now profited from the power of science. This increased his confidence. He was increasingly looked up to by his customers, employees, and most importantly for a listed company, he was looked up to by his shareholders as well. I think some of those early AGMs and shareholder meetings, there were hardly any questions, probing, pointed questions. Everybody was just applauding Dr. Reddy. And I must confess, one of my reasons favorable interest in Dr. Reddy was spurred by the fact that my father was one of the earliest shareholders in Dr. Reddy's laboratories. And naturally, he was a great fan. If you make a lot of money, you become a fan. Early success came with a burden of high expectations from watchful admirers. It gave Dr. Reddy an added sense of responsibility and the motivation to do things with impact. At the same time, he was developing a vision that was bold and expansive to bring affordable medicines to the masses. Ironically, the final push that gave impetus to his ambition came not from his Indian audience. It came from his multinational bulk drug customers. Unlike his audience in India, they expressed incredulity about his technical abilities. These slights to his pride made him determined to succeed and prove them wrong. It was no longer enough to supply multinational companies with bulk drugs. He now aspired to become an innovator of the first rank himself. But drug discovery is quite a different animal. It is complex. It is comparable to designing and building a passenger aircraft, perhaps even more complex. Dr. Reddy was ambitious, but he was very well informed, too well informed to be naive about the challenge he had taken on. Drug discovery was not about chemistry alone. He had to teach himself biology. For this, he relied on his network of friends, allies, and associates to climb a steep learning curve. His sources of counsel were an eclectic group of drug discovery executives from pharma companies, consultants, peers in the industry like Yusuf Hamid and A.V. Ramarao, other scientists, and employees. He enjoyed talking about his ideas, and listening to and reading about drug discovery experience of people was his favorite habit. He was a superb raconteur. I've heard him tell a lot of stories, and each story would have a telling message at the end. But even more importantly, he was a great listener. His approach to finding the right strategy was through long conversations with people whose opinions he trusted. He would use almost a Delphic method of asking questions. He would ask a probing question and listen very attentively. This served him very well. People opened up to him because they wanted to help him succeed. Dr. Reddy also hired solid people of character, and having hired them, he trusted them implicitly. In return, the people he chose to work with him would often go the extra mile when things got tough. But for every entrepreneur, for every unfurled sail, there is always a wind behind it. And I have to mention this point as well. This was true for Dr. Reddy. He chose a newly emerging class, the glitazones, for which at that time the molecular target was not known. Therefore, testing the molecules needed the ability to make gram quantities and feed them to rodents. My colleague, Dr. Rajagopalan, knows this. It is a slow and grinding process that even large companies with high throughput technologies found hard to do, especially when they had numerous competing priorities, competing with the same resources, that is, making gram quantities and doing in vivo pharmacology. This leveled the playing field somewhat. Within 3 to 4 years of the launch of the discovery program at Dr. Reddy's, a milestone was reached in the form of a licensing deal with a global multinational. The breathtaking speed of this accomplishment was unprecedented for any company starting drug discovery from scratch, especially on the back of a relatively inexperienced team of scientists. I think Dr. Rajagopalan was the only cart-carrying drug discovery development scientist in the team, and it was especially unprecedented for an Indian company of the time. Despite early success in the form of licensing deals, Dr. Reddy was attuned to the changes taking place in pharmaceutical innovation, a major transforming event, which was not as transforming as we expected it to be, but it was still transforming in terms of changing our mindset and our mentality, which was the sequencing of the human genome. And Dr. Reddy pivoted before pivoting became fashionable. He blessed the creation of a new startup company, of which I became a part, Origin, a drug discovery company focused, at least to start with, on structure-based drug design. He also founded Dr. Reddy's Institute of Life Sciences at the University of Hyderabad. Later, he founded a biotech startup, Carius. He was sensitive to the coming era of drug discovery, which was increasingly informed by a biological insight. Quite often, the biological molecule itself was a pharmaceutical product. The sequencing of the human genome, instead of simplifying things, has uncovered the exponential complexity of biological systems, some of which you just saw in the presentation by Professor Yona. These are actually veritable dancing soups of molecules, and you saw those molecules dancing on the screen there. At the same time, biology, on the back of powerful nanoscale and high-throughput instruments, has also become an extremely productive and fertile ground for innovation. If biologists had 1,000 topics to work on in the 80s, today they are chasing perhaps 10 times or maybe even 100 times more ideas. But with automation, robotics, and artificial intelligence, they are chasing them 1,000,000 times faster. This new knowledge holds promise for defeating many diseases that are commonplace today: cancer, diabetes, heart disease. The new biology also poses a challenge for companies, especially pharmaceutical companies, venturing into innovation. They are faced with a literal embarrassment of riches. So many targets. This presentation itself saw so many drug targets. If they pursued every promising idea, they would spread themselves too thin. So increasingly, the model is for pharma companies to collaborate with biotechs and with academia. The hottest hubs of biotech innovation in the world today, and it is not a coincidence, they are all clustered around leading universities: Harvard, MIT, Cambridge, UK, Stanford, Berkeley, UPenn. Just a few examples. Is Indian academia ready to play a similar role in innovation in India? I believe they are, or at least they can be, but they need our support. They need your support, my support, and all of us as Indian citizens need to wake up and support this ecosystem. Because helping academic science is a win-win proposition. There is no country in the world today that has raised the standard of living of its people in the absence of a vibrant ecosystem of science. There is none. Well, there are some, like the oil-rich countries. If you are rich in natural resources, you can lift the standard of living of your people without doing any science. But if you do not have natural resources, then the only way to lift the standard of living of your people is through science. The organization where I work, Ignite Life Science Foundation, has the mission to support the creation of a strong, vibrant, and productive academic ecosystem in India. I think Dr. Reddy would have appreciated the need for this intervention. In the 90s, when we were at Dr. Reddy's doing discovery research, where did we look for academic collaborations? Invariably, it was overseas. Northwestern in Chicago, St. Jude's in Memphis, NIH in Bethesda. These are the places that actually enabled our discovery at Dr. Reddy's. We need universities here in India to be beacons of research excellence. They must become the go-to places for innovation. This is the task for Ignite. We would like to accelerate the changes needed for transforming Indian research institutions into go-to places for innovation. But the question arises, how do we accomplish this? And I think one of the easiest and reflexive responses is to pour more money into research. I don't think that is sufficient. I think we actually need to start at a different place. We need to start by improving the quality of science we do in India. We need to make our institutions and our administrative mechanisms more responsive to the needs of our scientists. In India, like in most countries, the government is the single largest buyer of research, establishing what economists refer to as a monopsony, the opposite of a monopoly. There is hardly any competition for government funding mechanisms. This must change. It has to change. We must establish private sources of funding for science, reliable funding that is at the same time much more meritocratic and much more demanding of the quality of work done. Our mission at Ignite is to support scientists to create knowledge through high-quality science executed efficiently. The concept of efficiency is often not applied to academic science, but I think we need to start seriously thinking about how every rupee returns value to us when we invest it in academic science. When appropriate knowledge, when it is appropriate, must be coupled to market mechanisms that can take them faster into the marketplace. India is a place of enormous promise, at the same time, enormous risk. Promise because we have all the ingredients in place to succeed in innovating our way out of the challenges we face. Risk because the cost of failure will be punishingly high for 1.4 billion Indians teetering on the precipice of runaway growth and non-communicable disease. This is an agenda for our biopharmaceutical industry, but they cannot succeed, and I would like to emphasize this, they cannot succeed without a thriving local ecosystem of innovation that has upstream of it an academic research ecosystem feeding it with the high-quality knowledge that is the fodder for innovation. As a leader and pioneer in innovation, Dr. Anji Reddy got this. And the DNA of the company he created retains this spirit. So I was not very surprised when Prasad called me a month ago to commit multi-year support for Ignite in its mission of strengthening the academic research ecosystem in India. So on behalf of all of us in the room and all Indians in India, a very big thank you to Dr. Reddy's laboratories. Actions and events have both foreseen and unforeseen consequences. Over the years, many alumni of DRL, and some of you are in this room, have left and started their own companies. Who would have foreseen that Dr. Reddy was laying the foundation for what was to become the bulk drugs capital of India and the world? I cannot place a number on it, but I would wager that the value created by Dr. Anji Reddy for the country is far, far greater than the market cap of DRL today. I am sure that would have given him great satisfaction. Similarly, the seed planted today by Dr. Reddy's laboratories by funding Ignite's mission will, we hope, grow into a large fruiting tree of innovation. But beyond that, this philanthropic gesture is bound to create immeasurable benefits for the country through events we cannot even foresee today. We start small, but we will end big. If Dr. Reddy were here with us today, what would he be telling us? Something along the lines of, stay hungry, stay foolish? No, he was not a big fan of foolishness. Instead, I think he would have said something like this: be audacious, go climb mountains, the view on the other side is better. Before I conclude, I would like to repeat that quotation that you already saw from Dr. Reddy's biography in his own words, and he says, I strongly believe that drug discovery is a noble, perhaps even a spiritual pursuit. It is beyond bottom lines and investor relations. The mission is to improve the quality of life and life expectancy itself. This requires good science. But for science to be good, it has to result in affordable medicine. Indian enterprise is best positioned to deliver both. That is my unfinished agenda, end quote. We start today with an unwavering commitment to good science so that we can bring to completion Dr. Reddy's unfinished agenda. Once again, I thank DRL, G.V. Prasad and Satish Reddy for having faith in Ignite's ability to make a difference. You are joining us at the start of this journey. Please walk with us till we reach the destination. Thank you very much for listening.
Thank you very much, Dr. Swami, sharing your nuggets and remembrances. It almost felt like we had traveled back to the 90s and were experiencing the atmosphere and the excitement and the ambition of those days. And thank you also for sharing, some of the, I'll say, the crux of the problem today. While we are still looking for collaborations many a time to academic institutes outside India, we really want to expand much more in India as well. And thank you for initiating this approach. So thank you, so much for your lecture. I would like to invite Mrs. Anuradha, request her to come on stage to share, present a memento to Dr. Swami. Thank you very much. Thank you. So I would now like to request Mr. Satish Reddy to share his thoughts with us on this occasion.
So very good afternoon, everybody. It's almost getting to noon. First, I wanted to thank Professor Yonath for being here. Your lecture is a befitting tribute to Dr. Reddy, our founder chairman for Dr. Reddy's Laboratories and my father. It means a lot to him. It was a great presentation from an amazing institute, Weizmann Institute. This was an institute which my father used to talk about very frequently in his conversations because he had a special attachment to especially institutes of excellence. And he used to be inspired by them. He used to learn a lot from people. And also wanted to particularly thank Dr. Vijayaraghavan for being here. In fact, I had the good fortune of meeting Dr. Vijayaraghavan when he was a principal scientific advisor to the Honorable PM. From the industry, we used to meet him and always keep complaining about various challenges. But that was with the intention of smoothening the roadblocks, which came along, innovation, which again was very, very dear to Dr. Reddy. So that was the intention with which we used to approach him, and a lot of the things he gave us a patient hearing and paved the way for it. Also, thank you for your lecture today, Dr. Vijayaraghavan. It was great. Also, Swami, who's had a very long association with my father, he's also talked a lot about him as a person in terms of what he meant to the companies that he founded, the institutions that he founded. So thank you, Swami, and especially with his Ignite Foundation, we would definitely see a lot of great things coming out of that. So like Prasad talked about it before, and even Swami just before me said, I would always think about what if Dr. Reddy was in this room and he saw all of the people who are here, especially distinguished people, distinguished scientists. And I was just remembering Dr. Govardhan Mehta, because one of the conversations with him at some point of time, there was this, problem we used to have with enalapril. This was way back, early 90s, in Russia. The court case was being fought, and then it required a lot of scientific solutions in terms of presenting our arguments. I remember accompanying my father to the University of Hyderabad and spending time with him. And then the passion with which they used to discuss science. I was just thinking about that when I was in this room, because he would have conversations and he would especially be partial to scientists. There would be a twinkle in his eye. He would be very, very intense in conversations on science, pick up a lot of ideas, discuss various things, and that's where he would be. And I also remember one more thing, Dr. Govardhan Mehta, you used to mention that, especially at NCL, when he was there at the same time as you would know that Dr. Reddy was in that canteen because of his laughter. And that's a distinctive thing I think most of us remember, because I mean, even when conversations with you people, he would laugh a lot, right? Because he would recount a lot of instances and various things that would actually provoke his memory, and then he would talk a lot about it. I see Dr. Guru Reddy here from Continental Hospitals. He also recollected a lot of conversations that he used to have with him. And, so many people in this room, I mean, especially people, on the philanthropy side, the institutions that he built, nice foundation. Dr. Padmanabh Reddy is here. Manoj is from Nandi Foundation. Great institutions that he built. Dr. Reddy's foundation itself, which he started with. These were institutes because he always had this passion of giving back. That was in the video you heard him getting provoked, by that article in Newsweek, which talked about, India being in a situation which was worse than sub-Saharan Africa. That provoked him. There was a shareholder in one of our AGMs who also provoked him a lot, saying that, okay, you have earned a great deal for your shareholders, but what did you do to give back to the society? These were provocations for him on his quest for philanthropy, because he always, wanted to give back in a small way, in a big way, building institutions. That was something very, very close to his heart. So that's something, I once again thank all the people for making it here. Also a lot of people from Dr. Reddy's, who again have very fond remembrances of Dr. Reddy in the working years with him. Also wanted to specifically thank organizers for this event, Srinivas, especially Director of the Institute of Life Sciences. It was also befitting that Srividya compared this event, because she's again a senior scientist at Dr. Reddy's, process engineering, at our research group. I also now see Dr. Ramarao at this event. Once again, thank you for making it, Uncle. It means a great deal, because I know you since childhood, so you've been a family friend. It's something, which all of us relish. Also, like I said, I think Prasad mentioned this during this time. So in the conversations that we used to have with him, of course, generics business was not of interest to him. But then when he used to even speak about research, everything he used to speak, that we assumed that we knew everything that he spoke. And a lot of it used to be chemistry at that time. And even though being a science graduate, I had to go back and also brush up on some of the basics, because it took time to understand what he was saying. Because today is the world of Google. You could just Google and you could get the answers. But way back in the 90s, we didn't have the luxury of that. So just to get into an even keel with him on the conversations that he had to brush up on basics. But that was Dr. Reddy. He was very passionate about science. He was very passionate about the work being done in various parts of the organization, various parts of the institutions across the country. He always believed that we could always do more, and that's something we always remember, because he was very optimistic. He was very optimistic when he embarked on the drug discovery program. He was very confident that we would. For him. So today, marks the 10th year of his passing away. So we started the memorial lecture. So this series will continue. We would invite scientists and accomplished people from across the world to be speaking on an occasion like this. But just to give a flavor of what we normally do on this occasion every year on March 15th, again, something that was close to his heart, which was on philanthropy. So every year, we used to mark March 15th as a day of the spirit of giving to people across the company, people who have made a difference, people who created meaningful impact in the field of philanthropy. They used to come and talk to us in the company, and the people across the organization would then draw inspiration from them and also get inspired to give back. This is something we had done at the peak before the COVID started. Almost 3,000 people used to volunteer in this spirit of giving. This is something we have taken it across the organization, people who would be inspired, people who would want to give back, people who would commemorate that particular day, not just that particular day, but through the year to give back. That's something we used to do in the past few years. So just once again, in closing, it means a lot of us to the family that all of you are here today. At the institutions that he has founded, again, it means a lot to us. And I think to all of us, it means a lot that what we can do is to continue his legacy in science, in terms of the passion to keep it going, to excel and to innovate. Also in business, to also keep nurturing and also build on the values that he stood for when he founded Dr. Reddy's and the institutions that he founded. And also, most importantly to us, in philanthropy, to make sure that we create meaningful impact in the lives of people. And again, just to close, I would once again say that his dream of this unfinished agenda is something that we would pursue. And the seeds of the various things that we have done at various stages, not just in the company, but also in the institutions, it would lead to that, at least in this decade, and that would be the most fitting tribute to Dr. Reddy for all that he always dreamed of. Once again, thank you for all being here. It's great to have you with us. Thank you.
Thank you very much, Mr. Satish. And I'm sure all of us here are inspired to keep working towards this and living, making sure his legacy continues. I would now like to open this up for an interactive session where I would like to invite some of the distinguished guests, to share their remembrances and thoughts on this occasion. So may I call upon maybe Professor D. Balasubramanian, perhaps, to start this, or? Sure. Or, it's open, so we'd like to invite, any of the guests here today who would like to share. Professor Govardhan Mehta, if you can.
Well, everything that could be said about an outstanding, I would say almost iconic career that Dr. Reddy led has been said, and I cannot really match the kind of speeches which were made and tribute was paid to Dr. Reddy. I can only say that I don't know whether there are many people in this audience who knew Dr. Reddy as long as I had known, because we studied together, and we were together in 1964 and 65. And as Satish has observed very correctly, I could always identify in the place which we shared and lived together, a hostel, where Dr. Reddy is because of his laughter. I used to track him because of his laughter. And of course, our interactions continued. It so happened that we both landed up for some period of time in Hyderabad. And when I was in the university, he on many occasions called me and he said, I want to interact more with you. But I was in a different world at that time. And, I had my own agenda for academic pursuits and so on. We didn't interact much with him as such. But we used to travel very often together on a flight to Delhi. Even when I moved to Bangalore, we would even for a seat which is in the neighborhood, and we'll say, come there, we'll sit there. And of course, next thing what he will do is to ask the air hostess or somebody to get some napkins. And he will write structures on these napkins. He said, have you heard this thing? This is very, very good. Tell me how it can be done. This passion. Every moment he used to do. I wish, now I recall, I wish we had kept some of those napkin jottings together. Maybe there were some very good ideas there, but they never went beyond. So I'll not take much time. It's an occasion where I like to join everybody in paying tribute to Dr. Reddy. In fact, today's event is a celebration of a great life. A life of passion, commitment, success, and aspiration. All in one. You rarely get such combinations in everybody. So once again, to Satish, Prasad, family of Dr. Reddy, it's an occasion to rejoice. And I join you in recalling and rejoicing.
Sir, I request Dr. A.V. Ramarao to share a few thoughts, please.
So it is something great to me to asking me to say a few words. It all goes back during my BSc days at AC College. He was my junior. I know him at that time. When I moved to UDCT at that time called the present ICT, he followed me. We became very close during those days. And that was the time even the family became both of us, used to be very closely associated. And then when I moved to NCL, the next he asked me, why can't I go? I mean, he wanted to come back to NCL for his PhD. I took him to Dr. Doraiswamy, and Dr. Doraiswamy at that time not much known. So immediately accepted because we were BSc Tech students, not chemical engineers. But anyway, already I think Prasad, somebody mentioned about, I'm sorry, what I mean is he has exceedingly done very well because we are biology people and not the chemical engineers by nature. Only our chemical engineering was only peripheral, confined to unit operations. So that is one thing he wondered. Then he saw one advertisement from IDPL and came to me and asked me, should he go on? I said, why not? I know Mr. Rajarao at that time, who was working as a technical officer. I spoke to him about Dr. Anji Reddy, and that is the way he moved to IDPL. We were closely associated during that time when he started first time what is called Uniloids. I used to go regularly whenever I passed through the Guntur, the Hyderabad. I used to go to his R&D and used to spend some time. That was the way that my closely association. Then one day he called me when I was heading the organic chemistry at NCL. He wanted to make ibuprofen. By that time already ibuprofen I gave as a consultant to Cipla. So, as a scientist I can't pass it on the same information to him. Then I told him on phone, why don't you do a totally new approach? Starting from, no doubt, the same acetophenone, isobutyl acetophenone. You reduce it, take it to chlorination, and make to cyano and cyano to the acid. In fact, I didn't realize at that time, of course, totally it's new, and Dr. Reddy picked up all this and created what is called Cheminor at that time. And that was the way It was the first time exported, and there was a case. He could win the case because it is not the same what the boots were making. So what I mean is, closely interacted also with him. And that is where the whole thing. No doubt the organization has grown substantially. And I also interviewed Venkateshwar along with Anji Reddy to bring him into the picture. What I admire Dr. Anji Reddy, he was much more ambitious than anyone. And the second thing is, he started the foundation. And that is the way always he feels whatever we have earned, part of it has to go to the society. That was a noble idea which no industry follows, I must say. So finally, it is unfortunate he was all the time dreaming to discover a new drug. It has gone up to the almost phase 2 trials, or at the phase 3 it failed. So otherwise, that was the only dream which he has not succeeded. Otherwise, Dr. Anji Reddy is unique. The only thing, one thing I must say, Hyderabad would not have been bulk drug industry but for him. He was initiated, and also he trained several people. I don't want to name them. Today, they're all the big giants. And that is the biggest contribution of Anji Reddy to make Hyderabad as a bulk drug, and also for the country itself, as a reputation has gone up as what we call India is providing not only the bulk drug, is the pharmacy of the world. So the credit should go, people don't realize, should go to Anji Reddy. Thank you very much.
Request Dr. Venkatejasti to share a few thoughts.
Good afternoon, everyone. This is Venkat Jasti from Sugan Life Sciences. As everybody was talking, he's passionate about his drug discovery program. One day he called me. He knows that I am into some kind of a drug discovery program at that time, but he didn't realize when he started Carriers Therapeutics, he's working on central nervous system diseases. He was looking for support in doing things in USA. And somebody told him, why are you looking at it when your guys are next door to you, right behind your office, guys doing things for you? Then he called me. He said, I always talk to my son-in-law and son, why you guys are not concentrating, and I know, but I forgot about you. And at the same time, he was very passionate, and we used to have quarterly meetings on the developments that are taking place in central nervous system drug discovery. In one of the big meetings, as a chief, in one of our association meetings, he said, I know I'm crazy spending money for a 0 to 1. That means there is no success guaranteed in drug discovery, but I have money. But I see one guy sitting in front of me, he doesn't have money, but he still spends money on drug discovery. That is Venkat Jasti. That's the way our relationship goes, and until his death, we used to meet every quarter and exchange notes, and he keeps invigorating me in the discovery, which I continue to do it. Thank you. That's the greatest thing for me to remember.
It's truly a great honor to speak about Dr. Reddy. Dr. Satish and Prasad have very eloquently paid the tribute as family members, and several of you who all worked with him. It's my misfortune that I didn't have an opportunity to work with him, but nevertheless, I've stayed in the US for almost 3.5 decades, and it was my interaction with him first year in Hyderabad on my trips back from the US. He was invited to my home for dinner along with auntie who is here. She was a very dear friend to my mom. And at that point in time that I went to the US when I was about 23 or 24 years old, I did not know much about, Dr. Reddy's, but then, understanding the pharma industry, what is happening and whatnot, and hearing from people about his, his origin and his establishment, his drive, and a man of science and all of those things, for a young person like me, truly, had a tremendous impact that somebody coming from Hyderabad, somebody coming from India, was able to think on those lines beyond simply as an entrepreneur and as a businessman, and who has all the wherewithal with a foresight to put an amazing team in the future years, and a drive and a passion for him to think about that India should make its first drug in the country. It was during those times while in the US, I'm a gastroenterologist, hepatologist, and all of those things, and I was speaking for various pharma companies before the Edward Kennedy bill came in and squashed everything. So I had my decent first-class flights and business class flights, traveling across various states in the United States and speaking from Janssen Pharma to Pfizer and all of those things. And I was also an FDA researcher in the 1990s during the evolution of the hepatitis C drugs with the National Consortium in the US. So that was my foray into the world of science, of what really happens in terms of drug discovery and evolution, the pains, the difficulties. But I always wondered, being in a country like America, which is endowed with so much of wealth, of billions of dollars sometimes, and how could India really scale up to that? Is this really possible? On one hand, I used to sympathize or empathize, but then I thought Dr. Reddy would have this miracle. I heard that he opened his offices in New Jersey, in Georgia, and he has 450 PhD scientists. We all used to discuss in the US. I said, listen, somebody who is truly in the private world, manufacturing, generics and a lot of other things, is interested in putting 450 scientists together. I think there was a real purpose behind this man. And beyond that, a few times I had the opportunity to meet him when he visited in the US, particularly the American Association of Physicians of India conferences, the American Telugu Association, and very interestingly, probably is one of his last trips. The 38th or 39th Congress of American Association of India was held in Houston. I was the organizing secretary, and I happened to introduce Dr. Reddy for that year's leadership award in Houston. It was kind of serendipity and an accident that it happened to me in Houston, and I was there, and then I introduced, and when I visited India, I used to meet him. Sometimes I used to encounter him at Taj Krishna, or sometimes in other social events. He was a wonderful man, always inspirational, and he talked always in a very soft voice. And no doubt his laughter was truly his insignia at the end of the day. Those of you who worked very close with him. So I'm very glad, that a company that he founded will have its legacy rooted. And the Skions who are running today, will have been taking this, repeatedly to the next level. Perhaps I see, when I look at all the global companies, Indian pharma companies, Dr. Reddy's always strikes me as an extraordinary decent company which follows the rules of science, rules of business operations, and it is kind of held in a very high esteem. One last little anecdote. When Dr. Reddy's was trading in the US, several of my American patients, this is a true story, in the 2000s, they used to sit at least once a week. Are you kin to Dr. Reddy's? Then I used to ask them, no, I'm not kin to Dr. Reddy's, but I know Dr. Reddy's. We have invested in his company. That was the next thing, So many times, probably at least 100 times over a span of several years, I came across where American patients used to ask me about his kinship. So his legend was when his company was floated, and how many people kind of felt comfortable in investing and talking about him. He really put India on a global map. And thanks for this opportunity to share my thoughts. Thank you.
Thank you. We'll request Dr. Murli Ramchandra to share a few thoughts.
Thank you, Srinivas, for this opportunity. I'm Murli Ramachandra. I head the Origin Oncology, which is a subsidiary of Dr. Reddy's, working on innovative therapeutics focused on cancer. Around 2005, when I decided to come back to India from US, I was looking at multiple opportunities. I think it was largely because of Dr. Reddy's reputation as somebody who wanted to make a difference in science and innovative medicine. I think that attracted me to Dr. Reddy's companies. So I joined Origin in 2005. I was head of biology, then became head of discovery. So as part of Origin, I had an opportunity to interact with Dr. Reddy on multiple occasions. In the beginning, it was probably once a year or twice a year or so. But in last 6-7 years of his life, I think we were fortunate enough to have interaction pretty much on a very regular basis. On a monthly basis, he would come to our office in Bangalore. He would have, it was a very sort of, it was calendarized. He would show up at 11 o'clock. We would speak about for an hour. He would find out what all the project we are working on. He would get really excited. And he would talk about all his experience, share his learnings, and give his motivating sort of tidbits and see how things have changed, how things have happened in companies like Pfizer, Novartis, Eli Lilly. He would inspire us with all his experience. So it was truly motivating. I think when I recollect all those interactions, that's probably one of the best things that could happen. I think that made a lot of us to stay in the organization, continue to sort of work towards coming up with a global product that's coming out of Dr. Reddy's companies. I think we believe that, at least in Origin Oncology, now our focus is completely to discover novel drugs. And we have multiple products. Obviously, as you know, we all understand the chances of success is extremely low. But we believe that with the track record that we have, with the people that we have, with the talents that we have assembled, largely due to his vision and which is continued by Prasad, Satish, with all their support, I think we've been able to build a extremely talented team. And all of us, I think we look up to Dr. Anji Reddy's. I think even that what I saw yesterday, I saw that on LinkedIn. All these are truly motivating. We are truly committed. We believe that we are in a very right position to fulfill his unfinished agenda. I think that's what keeps us motivated, makes us do our best to come up with something that truly makes a difference to patients globally. I think that's why we are trying very hard. We hope, and I'm sure we will succeed with the support that we have within the company. Again, thank you for this opportunity. Thanks, Srinivas.
Thanks, Murli. Maybe I think we'll have Manoj, if you can please, Manoj Kumar from Nandi Foundation.
Frankly, it's more than a privilege. It's also a very emotional moment for me because it's been with him almost every day that he was in Hyderabad. It was mandatory that I reported to him morning and evening, starting with a KBR Park walk and ending with time in the Nandi office. Well, what I want to say is this was probably an extraordinary memorial day today. Apart from the fact that he would have loved to know that a Nobel laureate from Israel, from an institute he admired, gave the inaugural lecture. He always dreamt, apart from the idea of drug discovery from India, that Indians should continue to be Nobel Prizes. That was one of his pet dreams. If there was a moonshot idea, it was that too. Just the recognition that science could be taken to that level and more Indians winning it. So it's also fitting in that sense that we had a Nobel laureate for this event. So thank you so much on behalf of all of us who got you to come to Hyderabad and talk, and for Satish and Prasad to get him. I just want to mention the fact that he had a very scientific approach to philanthropy. And I would say he probably chartered philanthropy 2.0 in India. Because I recall when he decided to be the founding chairman of Nandi and the foundation that he set up in Hyderabad and got me to run it, the first thing he said was, we will use a scientific approach to philanthropy, and the essence of it was, we will figure out how to ask the right questions. And as long as we got the right questions there, we would find the answers, and often they will be simple ones. And that was the sort of DNA that we had in taking Nandi to scale. Every time we struggled with problems which were as varied as how can every Indian get drinking water, which is WHO standard, or how can you ensure that there is no child malnourished in India. And his most favorite pet peeve, which was, how can we ensure that farmers make profits? Because he always said that I was the product of a farmer. I was a farmer's son, and his pet story of how his father distributed, herbal solution he got from turmeric, and a turmeric farmer, how he created solutions for dysmenorrhea at that time and gave away it free. So all those meant that he said, let's make farmers profitable in this country, again through agricultural science. And it's every time we were down, it was his extraordinary laughter, which was the only energy that we all had. I have not known of any other entrepreneur, business leader who would spend almost 100 days a year investing with all the key members of the non-profit, as if he was discovering another molecule while trying to solve issues of poverty. It's been an extraordinary commitment and passion that we saw, and I'm so glad that he's remembered and his legacy is as much for philanthropy as it is for science. I'll conclude by saying that he'll be happy out there to know that Nandi Foundation has probably impacted over 7 million lives. We have a solution for drinking water. We were the largest non-profit to feed school-going children. And on top of it, created a model of agricultural science by which close to 1 million farmers, including in tribal Araku region, has come out of poverty. I should definitely pay tribute to the fact that he was instrumental in creating another extraordinary institution in Hyderabad. My friend Dr.Reddy is here, who agreed to run it, giving up his lucrative practice as a doctor, which is the neonatal intensive care for emergencies. That was something, again, that he championed and set up. I recall going to every person possible to raise funds for it. He was extraordinary in its humility and told that I have no shame begging anybody to join this philanthropy bandwagon, and has raised crowdsourced money from as many people to create NICE, which is yet another institution out here with cutting edge science looking at saving lives. Overall, I would say that a lot of us are working to compete with all at Dr. Reddy's to see will his legacy be greater in philanthropy or science, because we are right there to compete. Maybe we will try to get 2 Nobels for this country by serving society better. Thank you so much.
Thank you very much. Thank you everyone for sharing, so many fond remembrances and, thoughts from each of your individual experiences. I would like to thank the speakers, our distinguished speakers today, Professor Yonath, Professor Vijayaraghavan, and Dr. Swami, for making this, here for this inaugural Dr. Kallam Anji Reddy Memorial Lecture. We are truly, honored to have you here, and had the privilege of listening to you with very, really inspiring science and how we can actually amplify the impact of science. I thank each and every one of the distinguished guests here today for coming here to pay tribute to Dr. Anji Reddy. And please do join us for lunch, which is being served outside. Post-lunch, we know some of you have expressed an interest to visit the Founders Memorial. It is open today, and if you would like to visit, please contact the desk at the lobby, and we'll be more than happy to facilitate the visit. Right? Thank you very much once again. Please join for lunch.
When the night has gone and the land is dark and the moon is the only light we'll see. No, I won't be afraid. Oh, I won't be afraid. Just as long as you stand by me. So darling, darling, stand by me. Oh, stand by me. Oh, stand by me. Stand by me. If the sky that we look upon should tumble and fall, all the mountain should crumble to the sea. I won't cry, no, I won't shed a tear. Just as long as you stand by me. And darling, darling, stand by me. Oh, stand by me. Oh, stand now. Stand by me. Stand by me. So darling, darling, stand by me. Oh, stand by me. Oh, stand now. Stand by me. Stand by me. Whenever you're in trouble, would you stand by me? Oh, stand by me. Won't you stand now? Stand by me. Stand by me. softly leave, and it's me you need to show. How deep is your love? How deep is your love? I really need to learn, 'cause we're living in a world of fools, breaking us down when they all should let us be. We belong to you and me. I believe in you. You know the door to my very soul. You're the light in my deepest, darkest hour. You're my savior when I fall. And you may not think that I care for you, when you know down inside that I really do. And it's me you need to show. How deep is your love? How deep is your love? I really need to learn, 'cause we're living in a world of fools, breaking us down when they all should let us be. We belong to you and me. Ba-da-dee-da, la-da-dee-da, la-da-dee-da. And you may not think that I care for you, when you know down inside that I really do. And it's me you need to show. How deep is your love? How deep is your love? I really need to learn, 'cause we're living in a world of fools, breaking us down when they all should let us be. When will I see you again? When will we share a precious moment